In our next section, we'll be discussing the physiology of transporters of small molecules. In the next section, we'll talk about the movement across a membrane that's driven by transporters and ion channels. There are several mechanisms by which a small molecule can get across a membrane. For molecules that are nonpolar, they can simply diffuse across a lipid bilayer, and that's shown here in the example of the pale blue circles. Most molecules of biological importance, however, are polar, and therefore they need some sort of mechanism to allow them to cross a membrane. Often, these molecules have a favorable concentration gradient to cross a membrane, shown here in the example of the brown triangles, but because they are polar, they require some sort of transport protein to allow this diffusion to occur. This is known as facilitated diffusion. Other times, molecules need to move up their concentration gradient, shown on the right in the example of the purple squares. Here, these molecules are trying to move up their concentration gradient. This can only happen with the breakdown of ATP to release energy to allow this process to occur. This is known as active transport. This is in contrast to the two examples shown on the left, which are forms of passive transport which do not require ATP. Passive and active transport have certain characteristics that are important to know. Passive transport occurs down an electrochemical gradient, and therefore ATP is not required for this process. However, simple and facilitated diffusion have some characteristic differences. In simple diffusion, you do not require a transport protein. Therefore, this process is not saturable and is not inhibitable. By contrast, because a protein is required for facilitated diffusion to occur, it is a saturable process, number one, and number two, molecules that are structurally similar to the molecule of interest can inhibit the movement of the molecule of interest. In contrast to these two, active transport requires the breakdown of ATP. In addition, because active transport always involves a protein to get a molecule across a membrane, it will also involve a system which is saturable and inhibitable. Active transport can be broken down into two basic types. There is primary active transport, where the transporter itself is also an ATPase, and then there is secondary active transport, where the transporter itself is not an ATPase, and therefore two different proteins are involved. Further, within this category, there are two subcategories. Symport refers to the movement of two molecules or solutes in the same direction, whereas antiport refers to the movement of two molecules or solutes in opposite directions. The next slide shows an example of active transport. On the right is an example of primary active transport. This is the sodium-potassium ATPase, which is found on virtually all membranes in the body. This sodium-potassium ATPase will actively pump out 3 molecules of sodium for every 2 molecules of potassium that are pumped in, and in the process will break down ATP. One important feature of this pump is that it is asymmetric in terms of the number of molecules that are moved, and therefore it will induce osmotic shifts. One important manifestation of this is that during periods of ischemia, when ATP levels are low, this pump will be inhibited, and therefore sodium will accumulate inside of cells, and cells will swell because of this. This is one of the main reasons why cellular swelling is seen in the setting of ischemia. The next slide illustrates one characteristic example of this and one important utility of this phenomenon for diagnostic imaging. During a stroke, areas of the brain are ischemic, and therefore the cells that would normally be served by the occluded blood vessel becomes swollen, one can track the amount of swelling that exists by looking at the movement of water molecules in that area. Diffusion-weighted imaging, as shown here, is one way of assessing how well water molecules diffuse in a particular area of the brain. In this particular sequence, areas that are white refer to areas where water movement is inhibited and therefore cells are swollen, and this has proven to be a very sensitive indicator of cerebral ischemia. In addition to primary active transport, we have secondary active transport. This is shown on the left of this figure. Glucose, as shown here, has an unfavorable concentration gradient to enter this cell. However, sodium has a very favorable concentration gradient to enter the cell. Glucose can utilize some of the energy expended by the flow of sodium into the cell, and glucose can go into the cell along with sodium. This is known as secondary active transport. It's active because without the sodium concentration, glucose could not move, and the sodium concentration gradient was generated via an active transport mechanism. If one were to inhibit the primary active transporter, one would therefore also inhibit the secondary active transporter. As mentioned previously, there are two types of secondary active transport. We have co-transport, sometimes referred to as symport, and anti-transport, sometimes referred to as exchange. Shown here is a cell which has a primary active transporter on the bottom, the sodium-potassium ATPase, which pumps out sodium and pumps in potassium. On the left, we have a symporter. We have molecule X, which is moving up its concentration gradient, and it does so only because of the favorable concentration gradient for sodium. X, quote-unquote, hitches a ride with sodium to enter the cell. On the right, we have antiport. Y is moving up its concentration gradient. It can do that when sodium enters a cell. Again, sodium will release some energy as it moves down its concentration gradient, and that energy is used to pump Y out of the cell. This concludes our discussion of the physiology of the transporters of small molecules.